So I'm working in Matt Rand's lab, and his focus is on lizard coloration and behavior. This is something he's been working on since his dissertation in grad school, I think. It's like 1990, back when I was born, he published a paper on this. And so we've been working with 3 different color morphs, and if you see on the board, we have orange, yellow, and white. And so there are 2 different things going on with the lizards. First of all, they've got the 2 different colors, either yellow or orange, and then white is they completely lacks pigment. And then the white ones are different from the yellow and orange in that they are also melanized. And so there's 4 of us in the lab, and we kind of have each taken our own little track. When I applied, I said I really liked molecular biology and behavioral biology. And so Matt's lab puts both together. So I've told you I do sequencing and I do molecular biology. But the fun part is that it's actually correlated with different behaviors in the lizards. So the orange guys beat up the yellow guys, and then the white guys are pretty rare, so we're not sure how they fit in yet. But we're trying to find that difference in their molecular biology that kind of gives them more aggressive behavior or not. We can figure that out. It'd just be really cool to say, yeah, this switch happened here at this point, and now these lizards like are different. I don't know. I think that'd be cool just to figure out the answer.
I'm doing research for the chemistry department. I work with Dan Kremens, another rising senior, and I work for Will Hollingsworth doing physical chemistry research. Our big project we're working on for all of next year is we're looking at how a certain inorganic molecule falls apart when you shoot it with these really high energy lasers. So that's how we align the laser system to make sure everything's, because if it's not perfectly aligned, there's not enough power in here to break the bonds on the molecule. By seeing how this molecule falls apart, we understand how other types of inorganic molecules bind to each other and how they react, and just helps us learn a lot about a broader range of inorganic chemistry. Physical chemistry is what this type of research is. It's very, you can sort of define it as you spend a lot of time doing equipment stuff, as we've done for 10 weeks this summer, and then you sort of run data in 2 weeks when everything's up and working, and then you analyze it for another 10 weeks. So you spend a lot of time like with your hands building things as well as like with computers. But this is physical inorganic, so you're sort of mixing the 2 fields together, which is what I really like, the interdisciplinary nature of it.
So I've been working with Kayla to figure out what genes in the plant Camia Christa are responsible for controlling when the plant flowers. So we have all of that RNA in little tubes, and then we can run PCR, which is a way of you put a specific primer that you're looking for a very specific gene, and then you can see how much that gene is expressed in the RNA. And so over the course of 24 hours, we're seeing if that gene has higher, different, or lower levels depending on if it's been light for 8 hours, if it's been dark for 8 hours, and then we're seeing how that eventually controls flowering time. The whole point of researching Camia Christa is hoping to be able to plant it in a mixed prairie and eventually use it as a biofuel. And why we're researching flowering time is depending on where the plant flowers is an indicator of how much biomass the plant will eventually have. And so you can use that to determine whether or not it'll be a good biofuel. And so if you can make plants that have a lot of biomass, then you can use them and they make good biofuels.